Hello, everyone. I'm glad to be here in this wonderful presentation. I want to thank the organizers for a very good organization. And I'm an assistant professor at Penn State College of Medicine. And as a disclaimer, I have a consulting company, but it is not related to this research. And so probably many of you heard about the master transcriptional regulators of cell differentiation. So the small number of transcription factors can, even one or two of them can be used to reprogram a cell into another. Especially nowadays, you probably heard about stem cell research and stem cell medicine. So people are using this overexpressing or activating those regulators and reprogramming cells into stem cells and differentiated cells. So these are usually called master regulators, and most of them are transcription factor proteins. And identifying these transcriptional factors that are master regulators are important. As I said, it helps you to reprogram the cells. It's kind of a more exciting side of the bioinformatics, actually. Instead of, most of the time, you're just observing the phenomenon, what is going on in biology in the cell. Now you can have the chance to kind of manipulate it. It's more kind of fun and have more clinical implications. And the problem is how to identify these transcription regulators. And one of the approaches is using the networks, so gene regulator networks, as Saif Elumi talked about a couple hours ago. So we built gene regulator networks and identified hub genes as master transcription regulators. And building gene regulator networks is a difficult problem due to its nature. So it is, if you have a look, so you need multiple sources of information. So because when you say gene regulator, like usually mean transcription factor to target gene regulation. So which transcription factors regulate which target genes. And you need some certain information for that. First, you need the gene expression for the transcription factors and the target genes. Sometimes they are correlated, sometimes not. And it is hard to detect the small differences in the transcription factor expression because usually those genes are expressed at low levels, not very high, and the current methodologies, RNA-seq or other methodologies, may not be that much sensitive to those differences. So you cannot capture those differences. And even in some cases, they don't change expression during the cell differentiation, but the epigenetic landscape is changing. So they can find new places, new regions to bind, and they can be activated, even though they have a stable expression. So in addition to gene expression data, you need one more modality, which is chromatin accessibility, which is epigenomic signature, so that you can identify the cis-regulatory enhancers, cis-regulatory regions or enhancers, which are activated and which control the gene expression. Now, thanks to advances in the genomics, we have the single-cell multi-omics protocols. Maybe you heard about them already. They are very famous. People are using in new studies. You can profile transcription, gene expression, and chromatin accessibility or other data modalities in the same cell, which is very powerful. It gives you a very clear picture of the cell types. You can annotate using this. You can annotate the cells using the gene expression data and check out the differences between the chromatin accessibility and check out epigenetic differences. So this can also be used to infer gene regulator networks, and they are being used. So there are multiple, and it depends on multiple steps. So gene regulator network inference, as relies on few steps. The first one is a peak calling. Maybe most of you are familiar with. There are many tools to call peaks from ataxic data. This is well established. And the second step is more difficult. So how to identify those peaks or enhancers to their target genes? Because this is more difficult because there is distal regulation. So the enhancer regions can regulate genes which are tens of thousands of kilobases away. So you cannot rely on the nearest gene. It doesn't help you. So you can use some co-accessibility or co-activity data, but they are usually the accuracy rates are not that high, usually 60 to 70%. So it gives you somewhat vague information. And the third one is the transcription factor binding to the peaks or enhancers or promoters. This is well-established methods that for decades people have been using motif analysis. There are large motif databases, but the problem is maybe if you ever did this analysis, the motif analysis gives you some vague information. It gives you a possibility, actually. It's not a definitive answer. So it doesn't say that if there are 5 motif binding sites in an enhancer, it doesn't mean it will definitely bind that rare. It is kind of stochastic nature. So it is just giving some probability. So it is not conclusive. That is making this problem very difficult. So actually in this presentation, I am not just showing I will not just show as a rose garden. I will show you dirt and rocks. It is a bit difficult field, difficult problem actually. You will understand more in the coming slides. But actually one of the important problems in this field I observed over the years was that there are dozens of papers coming out each year, and they are claiming that they have perfect accuracy, but you cannot replicate or reproduce their results because the data sources are given, sometimes shared, but the pre-processing steps are usually not shared. So it is near impossible to replicate the result. Unfortunately, you think that bioinformatics should be easily reproducible, right? You don't need any materials or something like that. It should be free, but it doesn't work like that. And there's not a common definition of it. What is accuracy? What is the performance even actually? So to close this gap, actually, we published a review paper last year for assessing GRN inference methodologies so that we can set a standard, actually. What is good or what is not? What is acceptable or what is not? And there are 3 main issues that you should take into account when benchmarking GRNs. The first one is the obvious accuracy. Is your inferred network consistent with the real biology or not, which is very, very difficult to assess. And the second is stability. What I mean is your methodology too sensitive to the small variations in the data? You want your method to adapt to the data, but the small variations should not kind of cause it to diverge. The results should be somewhat consistent. And the last one is the scalability, actually, which is often discarded. Some people come up with some algorithms, methodologies, but you try to run it and you cannot get it in reasonable time because it is just tested in a few hundred cells for a few hundred genes. And when you apply on a real data set, it is just not giving in a few days. So that's also something to consider. So for the first problem, we built a single-cell multi-omic gene regulatory database. So everybody knows about the machine learning field a little bit. So you are familiar with that UCI machine learning repository, where you can find many data sets and try and test your algorithm with it. But in biology, unfortunately, we don't have such databases that we can kind of test our methodology and see their performance. So we tried to fill this gap by building this giant database. And we did 2 things, actually, mainly. We built a reference network or gold standard ground truth networks using ChIP-seq atlases or Systrom or ChIP atlas or other databases so that you can identify which transcription factors regulate which genes, or perturbation experiment data sets such as knockouts, knockdown, overexpression of transcription factors. Or we also use protein interaction databases, although they are not very commonly used because they are not tissue specific, but they are partially used. We have these for human and mouse. And we have a single cell data sets for the same tissues for human and mouse, but it's not just expression data, but it's also single cell ATAC-seq, ChIP-seq, CRISPR-seq, even CRISPR-seq and DNA methylation. So we have multiple tissues in this database that you can benchmark any methodology. And so using this data set, using the benchmarking method set there, we benchmark the current algorithms for single-cell multi-omic GRNs. Actually, this is a hot field now. For example, some of the papers, I can give you an example. Pando, Celoreco, maybe you heard about them. They are published in Nature. And Linger in Nature Biotech and C+ in Nature Methods. So this is out there. Everybody's working on that. But these methods are very complicated. Many of them are very complicated, very complex. They are well designed, but you know this is RAG curves, right? It is, if you do a random guess, you have about 50% chance to get it correct. But what we've seen is that, so there's a saying that all models are wrong, but some of them are useful. What we've seen is actually all models are kind of wrong, and some of them are worse than others, actually. So this is a difficult problem, not because they are badly designed, poorly designed. This is a very difficult problem. Even we don't know are the ground truths are real ground truths or not. But some of them are slightly better. You can say, for example, Linger, which is a deep learning-based algorithm, is slightly better about over 0.6, which can be in machine learning. Under 0.8 is really poor, but in here, we cannot even reach 0.7. And that's one story. We also checked resource requirements, whether these are scalable methods, such as how much time they require, how much memory. So Linger is based on deep learning, takes more time, but it is better than others. And the Pando and Celebracle methods are they are simple, usual linear regression methods, so they run quicker, but their performance is not that good. And another thing which we test is stability. So is the method too sensitive to minor variations to data with subsample cells and computed overlap between the results? And it seems that Linger also has a very high stability when compared with other methodologies. And we wanted to develop a new algorithm because these algorithms are not doing that much good. We are working on a new methodology. So all these methodologies are based on, almost all of them are based on regression. So how you can predict the gene expression from transcription factor expression. But we thought that we can try something different. So we tried a classification-based approach for GRN inference. So instead of, can we do it in a supervised way? So can we use some prior data to train a model and predict the TF to target gene expression? And we developed several features such as expression, peak accessibility, and also TF to enhancer binding potential. And we developed two features based for the TF to enhancer binding potential. One of them is number of significant motifs hits normalized by total hits in the genome. And the second is a more continuous approach, or we sum up the binding scores in enhancers and normalize among all the peaks. And the results were very good, and it's too good to be true, actually. So very high. This is what we expect, because this is you are benefiting from the training data, actually. You are using some of the data for training and other testing, and it is not generalized at this moment. So although it is pretty good, we want to have, because in most cases, you don't have the training data for your tissue. So now what we are working on is that, can we build a generalized model so that with that prior data, you can run into your algorithm? And that's all my presentation today. I want to thank my collaborators, lab members, and also administrative support.